What?
Hi there, this is Kristen Rossler, and we're going to go ahead and do the first part of, I think we're on week 6 or 7 in bio lab. And the first part of this lab is actually quite fun. It's getting students comfortable with pipetting. So we have what we call pipetting school, and my daughter is here to demonstrate. All right, so there are, of course, a series of different micropipettes that are available. So she's got one right here. You can see that this one has like a peach-colored top, and it actually says on there, do you mind showing that again, sweetie? Whoa, slow, whoa. So if we look at it, we can see that there is a number. There's a number range that's written. I think this one says, what, 10 to 100?
10 to 100.
Yeah, 10 to whatever that is. I can't see
It's 100.
It's 100. Okay, so 10 to 100 microliters, and so that's the range at which this one is reliable. For these sorts of pipettes, we typically are going to use the yellow-colored pipette tips. So my daughter will demonstrate, of course, how to put that on. Little tippy-tappy, tippy-tappy. I tell students not to become serial killers. That's just perfect, yeah. And then you pull it out. Yeah, look at that. So just a little tippy-tappy, not to become serial killers. We just want to kind of get it on. So that's what we would use for the yellow, or what do we say, the peach, rather. There's also these other pipettes that our students are going to use. For today's lab, I would stick to either the small ones, either the red ones or the green ones, or those peach-colored ones. Let me see if I can, there it is. So those are also smaller ranges. I think the red ones go from 0 to 10 microliters, and the red ones also go from 0 to 10 microliters. So it just kind of works easier to just have them using the small ones today, since that's predominantly what they're going to use. Down the road, they're also going to be using the larger ones that go up to a whole milliliter. But again, I would just encourage them to use the smaller ones. For the red ones, and I think the green ones, they have to use the red pipette tips in these little boxes over here so that they actually fit. All right, so for pipetting school, they need to get used to how to hold the pipette. So of course, my daughter is demonstrating it quite nicely. Can you put your thumb on the top part, sweetie? So you can swivel that little white thing to the left or the right, depending on if they're left-handed or right-handed. My daughter's right-handed, so she's going to put her thumb up on the top. Right, and you can see how it kind of sits comfortably in her hand. What you would do to change the number, change the volume that you're pipetting up, is that sometimes there's a break on it, this white collar. Do you see that white thing, Zosha? Pull that pull that up, and that releases the brake. And now you can actually use that to swivel. So you actually, the white part, put your hand on that. Yeah, turn that around, and it shows how the number is changing. All right. And so for these, the numbers are all in black, you can see here. And so those are all microliters. So so here she's at, let me see, can you show it again, sweetie?
Oh, should I put it on?
Yeah, put it on, what is that?
This is 14.
Yeah, so it's at 14 microliters. So I usually start them off with 10 microliters because that can be utilized on both all the pipettes, the small pipettes that they're using today. So when they get it to 0, 1, 0, or on these other ones, it might also, there might be a green number. The green number is after the decimal point. So for instance, let's see if I can pull this up. Yeah, thank you.
Is there not a
let me see the numbers on the side. Yeah. So here you can see that there's a decimal point. And so Zosha is scrolling it down, and she's going to get it to 10. All right, so they're pretty intuitive. The red ones, hopefully you can figure that out. Again, it goes 0 to 10. So yeah, so that's how you do it. And what I do is I start the students off on just kind of getting comfortable with them. What I typically do for this part is I get some paraffin film. So there's some paraffin film that's usually kind of floating around over on one of the benches. Yeah, and I just kind of cut a piece of it. And there's obviously a waxy kind of hydrophobic part, and we're going to have that facing up. Yeah, perfect. And so we have some food coloring that's floating around in the labs. Usually it's in one of the cabinets. Yeah, and so I just kind of put some of the food coloring out in so that each group can have access to it. And Zosha's going to demonstrate. So I dare say you've pipetted before, but let me just kind of go through it. You might remember that when you push down on that top plunger on the very top part of it, there's kind of two breaking points. There's when you push it down, you're going to feel the first stop, and then if you push it all the way down, you're going to feel that there's a second stop. So the trick, of course, with micropipetting is that, yeah, do you mind getting everything out of that first? Good, okay, good. So she's going to demonstrate. She's actually a pretty good pipetter. So when she wants to suck up 10 microliters, She's going to push her finger on the top plunger until she gets to the first stop, because that's going all the way down to the 10 microliter point. Then she sticks it in the solution, and then she's going to slowly
give it a
draw.
go?
Yeah, go for it. Yeah, she's going to go to the first stop, stick it in, slowly pull it up to the second stop, right, or just all the way to the top point. Good, and now she has exactly 10 microliters. She's going to go over here to the little hydrophobic, and now she's going to go all the way down to the second stopping point. That second from the first stopping point to the second stopping point is actually a little puff of air that comes out to help get the exact 10 microliters, the volume that you've set your pipette for, out. All right, and so what I typically do is I give the students like a big little rectangle of this and tell them to go crazy. We want to have uniform sized 10 microliter little dots of food coloring. They kind of like it. You can also talk about the why the water kind of beads up, and again, it has to do with the non-polar nature of the wax paper that we're dealing with here. So yeah, Zosha's doing a great job. So we do that for a while just so they can get comfortable with it. Then we move on to the actual lab portion of the experiment. So if you look in the notes that are posted in ULearn, hopefully you're going to see that it's the virus detectives. It's kind of funny, but it actually is a nice lab. What you're going to see here is that in the background material for the lab, I tell them a story. I say, okay, we've got this virus. This virus originated, and you work for the CDC and the Center for Disease Control. And initially, and this is actually based upon kind of a true story, is that there was I think it was the flu virus that started a really bad flu strain was originating, they found out, in Alabama. And here's the Alabama viral strain, DNA strain. And it was identified that these people were getting quite sick and having, pretty low mortality, or, many people were dying actually from this viral strain. A little while later, a very similar viral strain popped up that was similar to it in, I think it was in Pennsylvania. All right, so we had a strain in Alabama of a pretty deadly flu virus, and now we have a strain that's very similar to it in Pennsylvania popping up. And the CDC, of course, went in and took samples of each. It was identified, actually, that these two different strains were a little bit different genetically. This strain, the mutations that were in it obviously were a little bit more deadly, whereas the Pennsylvania strain actually caused sickness, but people recovered actually at a pretty high rate, right? Then there were people in Missouri who started getting sick. So the CDC is trying to determine, what is this a novel strain of the flu, or is it more related to the deadly strain in Alabama, or is it related to the benign strain that's in Pennsylvania? So this is again Missouri. So Missouri is kind of in between. So we kind of talk about obviously the geography and migration of people and spreading the virus, and what's a quick and easy way for the scientists to be able to determine what is the yellow, what is the Missouri strain. And what they did, of course, is they did PCR. So they amplified the viral particles, the viral DNA, and so they did RT-PCR actually, because flu is a retrovirus, but they did RT-PCR, developed DNA that was comparable to, of course, the RNA sequence itself. And then what they did is RFLP analysis, right? So restriction, fragment, length, polymorphism. And that's just where you use restriction enzymes to cut the DNA in a kind of a particular pattern. And we know that leads to essentially like a fingerprint, right? A fingerprint of a virus or of a specific piece of DNA. All right, and so if I remember correctly, when you do the RFLP analysis of the Alabama strain, I think you get like 2 bands, right? Whereas when you do the RFLP analysis for the Pennsylvania strand, I think you get 3 or 4. And so what they're trying to do is, and what they're going to figure out is that this is actually very much is actually identical in terms of its RFLP analysis to the Pennsylvania strand. So luckily the people are going to get sick, but yet they're going to recover at a high rate like the Pennsylvania. It's kind of fun. At least that's what we predict, right? How they're going to figure that out, right? So I tell them, we don't know, but we're going to go ahead and do an experiment. So we've got samples. These are the samples that have already been prepared. So again, what's happened here is the flu strains have undergone RT-PCR, so that the RNA was converted into the DNA of the virus. Then they were amplified with PCR to make more of them, obviously. And then they were cut with these restriction enzymes. And again, this is what we finally have. And we're going to run these different sequences, these different samples out on a gel electrophoresis. Many of them have not done gel electrophoresis before, all right? So just to remind you, if you haven't done it either, it's just we use one of these chambers. This is a gel electrophoresis chamber, and the gels have already been prepared. They contain agarose that was melted and put into essentially like a cassette, a mold. This is actually what the mold looks like. You usually tape the ends. Ooh, thank you. We put tape sometimes on the ends, and then we put, while the agarose is molten, we put this little comb so that it actually has wells that form in the gel itself, right? So the gel, we kind of put it in there molten. It's going to solidify. I tell them it's just like Jell-O, right? So if you've ever made a Jell-O mold, this is exactly kind of what's happening here with an agarose cassette. So they're going to get a gel that looks kind of like this. If the students want to touch them, then they can, because we're not actually using any of the ethidium bromide that sometimes is kind of a carcinogen, right? And you can see those wells, those holes. The holes don't go all the way down to the bottom, right? They go just a few micrometers above. So there's just a depression there, not a hole that goes all the way through, and that's kind of important. So we put the gel in the electrophoresis chambers like so. You always want to have, of course, the wells towards the negative end, the anode black end, because again, DNA is negatively charged, and it's going to be attracted now towards the red cathode, the positive end, as we kind of pump electricity through it. All right, that's kind of the basic premise of gel electrophoresis. So what you're going to have the students do, you can see Zosh is having a delightful time making this. Zosh, do you mind if I switch to you being the videographer? You just put down, yeah. Thank you so much. All right, good teenager. All right, so when the students are actually ready, so just to remind you, I think you know this, when you need to pop off one of these pipette tips, you can just use this little clicker thing, and it just kind of pops it off. There's
What
a little
clicker
garbage.
thing?
This little white thing, yeah, that little thing is the ejector.
Okay.
And that, rather than touching, obviously, the sterile pipette tips. So we're just going to get a clean pipette tip for each of the 3 samples that you're going to run out. And what we're going to do is that on the, do you mind coming over here, sweetie?
Yeah.
So what we're going to do for each one of these is, let me see. Yeah, let me get this. So I give each group, I kind of do this with each group independently. All right, so what I tell them once they're comfortable with how to use the pipette is that they're going to use, I give them one of each of the 3 samples, right? So they're going to have Alabama, Pennsylvania, and then Missouri. So it goes red, green, yellow. And we're always going to do it from left to right just so that we don't mess up. All right, so you're going to see either in the caddy for this or in the freezer for this, there's something called the UV view dye, right? So this we need to add to each of these samples so that it obviously weights down the fluid, and also that it actually impregnates the DNA so that it fluoresces, right? There's a fluorescent dye here that's going to bind to the DNA strand so that we can see it on the UV light box. All right, and I have them wear gloves for this. I don't have gloves. It's nothing really carcinogenic in here, so I'm just going to do as I say, not as I do, right? And what I am, I've done this for so long, I'm not super, there's some shortcuts, and so I just teach them the shortcut. So what I have them do to start off with is I just have them suck up about 10 microliters worth of this, right? And now I'm going to put like 3 kind of equal little dots. It's really that simple. It doesn't need a huge amount, and I just kind of add to it. So I've got 3 little dots of my dye, and that is usually more than enough to work properly. And again, I get like a little eject this, right? And so now what I'm going to have each of the students do, and I have them do it, take turns, is that they're going to take about 10 microliters, or they can take even less actually, but of the different samples. This one is kind of empty. Let me get them more over here. There's a little bit more of that. And they're going to get about 10 microliters worth, right? And so they would get their 10 microliters, right? And then they're going to come over to the first bead, And this is the part that's kind of, so this is the way I do it. I mean, I did this stuff for some good chunk, all my 20s and a good chunk of my 30s. So I just would put it into that, and then I want to mix it up a little bit, right? So I just go up and down a few times and kind of get it out. And now I want to suck up at least, the 10 microliters worth. So I just kind of put it into that drop, and I slowly pull it up, and I try to tell the students not to get any air bubbles, just because the air bubbles are going to make it kind of gross. And then what you do, and if you want, they can do some samples. There is some loading buffer. That actually might be a good idea for them to do initially, is that there's just some practice loading buffer. You might want to have them all do a practice lane before they even actually load the DNA. Hopefully that makes sense. So if I were them, do you mind coming over here, sweetie? So each group is going to have, each lab group is going to have their own gel. And you can see here that there's a myriad, there's like I think 8 lanes or 10 lanes here. I would have them do some practices over on these lanes, right? So I would have each one of the groups practice just with some loading buffer, the practice loading buffer that you'll see. And if you ask Tracy, she can get some for you. And just to put the 10 microliters in there, as long as it has the blue dye has enough glycerin in it that it'll weigh it down. And the blue is just so we can see it a little bit better, right? So yeah, so I would have each person in the group do at least one practice lane, all right? And then you're gonna have them, when they're actually ready to do the sciencey part, their experimental part, do what we just did, get them to mix the DNA sample, right? For instance, we're gonna start with Alabama. They're gonna mix it with specifically this one, the UV dye that allows it to fluoresce under ultraviolet light. And then they're just gonna come over here, and you kind of see, sometimes if there's a student who's done this before, and I often will demonstrate it for each group. An easy way to get students kind of comfortable with this is to tell them to put use both hands, and to put their elbows actually on the table, and they're just gonna lower it in. So I always joke with students that those students who were good at that game operation as a kid are usually really good at this. Then they just wanna really slowly release the dye, and it should fall into that well. I had a little bit of regurgitation there. That's literally what it's called. And most people are going to get a little bit, but the slower you do it, the less regurgitation and the less loss of sample that you have, right? So it looks pretty nice. So we're going to have that one's done. We're going to click off the tip, and then we would do the same thing now for Pennsylvania. We get about 10 microliters. I'd mix it into a small little bead there, mix it up and down, suck up the 10 microliters so that there's no air bubbles, and now I'd go to either that second lane. If you want, they can skip a lane if they have enough room, depends on how many people are in their group, etc. And yeah, you just put that in, all right? So hopefully that makes sense. So they would do, again, 3 samples in totality. I, again, try to have them do it all in the same order so that we know what we're looking at. So that's that. When they actually need to plug it in, kind of fun, right? When everybody's got everything plugged in, they're going to put the lid on it. All right, so we're going to set up the electrode system, complete the circuit. All right, so the buffer, I should have mentioned that. The buffer that I've put, it has to be covering the gel, right? So if you look at it from the side, thank you, Sophie, I'm really good at this. So we have the DNA, the agarose gel, and then we want to pour, you have to do this before, obviously, you load the gel, but I put the buffer all the way up. You don't want it so high that it's overflowing, obviously, but you need it to cover all the wells and cover the gel completely. It needs to be submerged, right? So that's important. The buffer, Tracy has some out. You can use this for multiple lab sections. In reality, we reuse TAE buffer all the time, unless it's for something that we're going to purify the sample from, and we're not doing that for this. So this is just a salty solution, and it helps to conduct electricity a lot better. You can remind them that we did that in lab 2 or 3, where we talked about how conductivity is related to the saltiness or the ions that are present. So then when they're ready to turn it on, they're gonna take this, or you can do it. Make sure the negative goes into the negative terminal, the positive goes into the positive terminal. Brilliant. And push it in. And I have them do it at about 110. You can crank it up if you want. There's some of the other systems that you can actually change. And once it's plugged in, actually ours is not plugged in. Let me just plug it in real fast. All right, it should fire up. All right, so we're getting 110 volts, And then you can show them that this is actually working. The electricity is being conducted through it. Of course, we have the negative here, and we can see those bubbles. That's how you kind of visualize it, that this is actually working. And what they're going to see over time is that blue dye, again, it's negatively charged, is going to be attracted to the positive red end. And so it's slowly going to start migrating. Hopefully you realize, and I'm sure you know, that the rate at which this migration occurs is proportional to the speed of the molecules, or the molecular weight rather of the molecules. So the speed, the smaller the molecule, the faster it's actually able to kind of get through all those little nooks and crannies in the agarose. And I always tell students it's like a person on a moped or a motorcycle in highway traffic, right? They can go wiggle through. Whereas the larger, bulkier products are not going to migrate very fast, right? It's kind of like the semi-truck on the highway, right, during traffic. So that's it. So I let it run until the blue dye, it's usually about 2/3 of the way through. So if you can get that blue dye to right around here, that's a great spot to stop it. It takes a while, obviously. So you want to try to get this set up as quickly as you can during the lab. I encourage then all the students to pull up the lab template for this lab and start working. They often can get this lab pretty much done before we even get the results. So when the results section, let me just show you how to do this real fast. When you're ready, when it's run about 2/3 of the way through, obviously turn off the electricity because the university frowns on us electrocuting students, right? And you're going to pull off carefully this. There's usually some backsplash that occurs, all right? And you can actually see the DNA has already migrated. The blue dye has already gotten into that gel a little bit. So what you're going to do then is, with gloves, they should have gloves, but again, do as I say, not as I do. Maybe I should grab some. Let me go and grab some. So what they're going to do is they're going to carefully carry over their electrophoresis chamber, or what I often do, just because other students spill it a lot, is I tell them to pick it up kind of like this, right, and kind of drip it, and then make sure their hands are on both ends because this is really quite slippery. And then they're going to come over, thank you, sir. They're going to come over to this cute little machine that we've got over here in the lab. This is a UV light box. Many labs that they would work in would actually have a room, a dark room, that you would do this in. And again, when students are doing this, it's a good idea for them to have a cell phone with them because they're going to need to take a picture of their results to include in their lab report. So we have one person who has at least their cell phone in each lab group, and they should all have goggles on technically, because this is a UV light box. The goggles are of course over in the back corner of the room, and you can just grab a pair there. Yeah, just right. High quality filming. It's awesome. Oh, that was really good. All right, so what they're going to do with their goggles on, and I should have my goggles on, but I don't. I'll be all right first. It's really for such a short period of time. I think we'll be okay. So they're going to slide very carefully their gel out as so, and then you're going to click the button on the side so that it's hopefully turned on. And then the trick with this machine over the years has been that this little button over here needs to be pressed down. Let me show you. Let's see. Is it on?
There's a green light on.
Is the green light on? Okay, cool. And so I typically will turn off the lights so that it gets a little darker. And then we sometimes, if you want, there's those black cloths that we used in last week's lab. Oh, so this one's turning on. There it goes. The black cloths that were used in last week's labs, you can kind of drape them over the students. And what you can do is just put this down, but this metal piece has to be pressing down that button right there, right? So you see how it kind of presses down, and we can start to see that the light emerges. And you know, we didn't let this one run very much, and it's kind of dark in here. There's a little fluorescent band right there. I don't know if you guys, yeah, it looks pretty good there. So that's actually the DNA. And it hasn't separated yet, right? What we should see is that there should be several bands that form here, but again, since I didn't let this run very long, obviously it didn't run. So what should emerge from that, and they're gonna take a picture of that, I actually have a picture up online, I think, of a good one, should everything go to hell. And again, so what we should see is that I think there's like two bands, I think, for the Alabama. There should be like 3 or 4 bands or something like that for Pennsylvania. And what we're going to see is that for the Missouri, the fingerprint for it, the RFLP analysis, looks identical to that of the Pennsylvania strain. So based upon that, we can conclude, of course, that the Missouri strain is identical, or similar at least, seems to be more like the Pennsylvania strain. All right, when the students are done with it, there is just some, these, we usually don't put them in the garbage can. We're just going to have like a beaker out here that they can kind of put them in, and we'll make sure that Tracy is amazing at making sure those get put away. All right, hopefully that helps, and I think that's just about it. Thanks so much.